Hello, you are most welcome, ladies and gentlemen, to Real Impartation Moment on Tuesday Night Anatomy with Daniel Okwan. Today's section, we want to look at, post-fertilization changes that will occur to the conceptus. Very important. That's what we want to see, leading to, implantation. Now, one important thing that I want you to be aware is that, yes, I told you that the spermatozoon, you may have about 200 to about maximum of 300 million, produced, per ejaculate, okay? So for this reason, that is for the normal, man on average. That's what we want to see. Well, some individuals may have oligospermia and all that, but we are looking at the normal case scenario. Now, I told you that the vaginal environment, is very hostile, and for this reason, most of the spermatozoa, will be sacrificed along the way. Studies have shown that out of these millions of spermatozoa, you may be having just about 300 to 500 of them, reaching the vicinity of the secondary oocyte. Yes, that's one thing that I want you to know. But my friends, the important thing is that I told you that the one which will be very fast, to fuse with, of course, so that its plasma membrane will fuse with that of the oocyte, secondary oocyte, is the one whose nucleus will be released into the cytoplasm of the secondary oocyte. That's what we looked at. Yes. So one thing that I want you to know is that before you can actually be fertilized the secondary oocyte, then there's one more thing that has to take place, and that is something we call capacitation. I believe this is not the first time I'm mentioning capacitation to you, because the important thing that I want you to know over here is that the spermatozoon will be covered with some kind of glycoprotein in there. So for this reason, that capacitation, to gain that functional capability to fertilize the secondary oocyte, you gain that kind of capacity inside the female genital tract. Precisely, I told you that it's going to take place right in, of course, talking about the cervix, talking about the uterus. Then importantly, it will ultimately take place in, of course, the isthmus of the uterine tube. And my friends, whenever I talk about the uterine tube, I am talking about the fallopian tube you know of. Okay, I'm talking about the oviduct that you know of. Yes, that's what we are talking about. Something because it's trumpet shaped, we also may call it sapiens. Okay, that organ that is trumpet shaped in females. Apart from, of course, the eustachian tube. That's what we look at. Okay, so now this secondary oocyte has been fertilized by this this functional spermatozoon. Now remember, I told you that the fertilization process is not just a single event, one step process that you are done. No, I told you the series of them, which we reviewed. So my friends, if you not watched that previous video, kindly get that last week video that we looked at, so that you will get complete understanding regarding whatever we are going to do today. Now my friends, take it at this point that, assuming this is, let's say, schematic representation, let me just say a schematic representation showing us, let's say, the assuming one portion of the uterine tube. I'm trying to show you, and let's say this is, of course, the womb, the uterus, where implantation is supposed to take place. Now, one important thing that we've seen, from last week, is that we will have the oocyte membrane. Of course, I told you that the oocyte plasma membrane, the oolemma, will be fused with, of course, the the vitelline membrane or the vitelline layer, okay, prior to being fertilized. Then after fertilization, there's going to be that kind of, broken of the bond between them. Okay, we've seen those things. But my friends, what I want to show you here is that I'm interested in telling you that still the zona pellucida will have to be intact. Okay, the important thing is that now we will see why the zona pellucida will have to be intact. Okay, it shouldn't dissociate prematurely. And so I'll have this zona pellucida. I told you it is this kind of glycoprotein, belt, which is non-cellular. Okay, so I have my zona pellucida over there. Yes. Now, one thing that we will see is that once there's that kind of fusion, okay, we will see that, assuming, let me just say, we'll be having this and then this. Now, if you followed what we've been talking about throughout this whole series that we started, you get to know that I told you that for oogenesis in females, for it to be complete, then we need a viable spermatozoon to, of course, fertilize or to fuse with, of course, this secondary oocyte we are talking about, so that they can complete its meiosis 2, which was arrested at metaphase. And so for this reason, once there's that kind of fertilization, then that's the stimulus for completion of meiosis 2. Okay, in, of course, the secondary oocyte, so that then they will form the ovum. Now, once we form the ovum, the nuclei over there, so I take it that this is the nuclei of the spermatozoon, and take it that this is the nucleus of the ovum. These nuclei, they are now going to fuse again. The real fertilization, the final stage of the fertilization, that's what we are in now. And that is what we mean by fusion of the male and female pronuclei. So for this reason, these guys, we call them pronuclei. And that is why we believe that when we talk about fertilization, ultimately, we want to ensure that there's fusion of the male and then the female pronuclei. Yes. And once they do that, yeah, they will come very close together, and then of course they will fuse. And therefore, we will form a zygote. Now, there's one thing that I want you to be aware. In other mammals, yes, we see zygote, true zygote. That's what we see. But in human beings, my friends, we don't really have a true zygote. Why? Because immediately they are just about to fuse, okay, then they begin to divide. For this reason, we can, yes, crudely say that zygote in human beings, which we are saying that it is formed from the fusion of the male and female pronuclei, yes, which is totipotent, in other words, capable of developing to become a full-blown, organism. Okay, we can say it is an undifferentiated two-cell stage. Now, it begins to divide. Now, there's a single, take it that there's a single cell, but it is not a true single cell. Why? Because, of course, it is not, fully separate, having fully separate, nuclei. So it's going to undergo that kind of division. That division is what we call cleavage. Now, my friends, I've done some of these videos, okay? I have them. Yes, I've done the female reproductive tract. We looked at the various changes, okay, along the uterine tube, okay, where we have more cilia, where we have more cells, which are going to make some, products, which are going to nourish this kind of conceptus by way of the zygote over here, and all the division products. So you can find all those videos. But in any case, I'll be taking you through all those dynamics, okay, that will occur over here. But today, I just want you to understand that immediately after fertilization, which we are not really getting a true zygote, it's not really a true unicellular cellular entity, okay, then immediately begins to divide. But my friends, the key thing I want you to know over here is that we still have a non-cellular belt over here of glycoprotein by way of zona pellucida. Yes, I told you that the zona pellucida, one of the things that it was doing was that it was ensuring that the sperm, because there were some kind of receptors over here, so that the spermatozoa can interact through some kind of acrosomal reaction. We explain those things for you. The induced acrosomal reaction. That's what we're talking about. Now, for this reason, that division, we are talking about division taking place, and this division is a mitotic division, not meiosis. It is mitotic division, and this time around we call it cleavage. Mitotic division by way of cleavage. Now then, the point is that why don't we simply call it mitosis? Now my friends, this type of mitotic division is not an ordinary mitotic division. The reason is simple. Now usually when a cell undergoes mitosis, okay, going through the M phase, there's also, that period, okay, very long period of rest, which we call it the interphase. And at the level of the interphase, that period, there's that kind of growth. Okay, there's that kind of G1, okay, S, G2 phase. That growth phase is present. That one, we don't see it in the case of cleavage. Although there could be an interphase, but there's no G1 and of course the G2 phases. No growth phase. For this reason, for every successive division, mitotic division, these cells over here, they will be reducing in size. I'm going to show it to you. They will be reducing in size. They will become smaller and smaller. Yes, that's one point. Okay, so there's no period where it is going to grow. It will be reducing of course in size. Okay, so that is the cleavage division that I want you to be aware of. Now again, because there's no interphase, it makes the process very rapid, quick, mitotic division. That's what we see over here in the zygote. Unlike in other eukaryotic cells, where of course we said that those somatic cells, they will undergo mitosis, and then of course that one it is very slow. For this reason, in the case of cleavage, the oxygen consumption and the energy that we need is more compared to that of the ordinary mitotic division. That's what I want you to know. Okay, for now. Now there's one more thing I want to put across over here, and that is the fact that this type of cleavage, we call it holoblastic cleavage. Now let's put this point down. The type of cleavage that we experience over here, we call it holoblastic. Now, which is opposite to another one we call meroblastic. Now, I'll be explaining these things for you. So it means that when we talk about cleavage, there are two things that I want you to know. Two kinds, main kinds. Okay, I won't go into the nitty-gritty so much, but there are two main kinds, and one of them, is what we call meroblastic. Meroblastic, we see it in reptiles, we may see it in birds, also in some fish also. Yes, they will be present over there. That is for the meroblastic. Now, if we are going to undergo meroblastic, now meroblastic cleavage, is synonymous with something we call partial cleavage, partial or incomplete cleavage. It therefore tells you that if it is holoblastic, then it's going to be complete cleavage. Now the question is why? The reason is simple. In the case of meroblastic, they have, those kind of, these kind of zygotes, or those kind of fertilized eggs, very yolky. In other words, the quantity of yolk, yes, which of course, which can be nutritive source, for this, for fertilized egg, is the one which determines whether or not we are going to have what we call, meroblastic cleavage or holoblastic cleavage. Holoblastic cleavage, where of course we see it in mammals, we see it in of course some amphibians also. Yes, that's what we see, holoblastic cleavage. Now even studies have shown that even of course these kind of flatworms, and all these earthworms, they go through this kind of holoblastic cleavage. Now that's what I want you to be aware of. Now, if it is meroblastic, then it means that the quantity of yolk in there is very large. Opposite to that, we find in the case of holoblastic. Now, because the quantity is very large, then we are not able to go through complete division. But if the quantity of yolk, okay, is small, in the case of the holoblastic case, then what is going to happen is that, yes, we can easily go through it. Yes, that's one thing I want you to be aware of, okay, for now. Now, in the case over here, which we are saying is holoblastic, one thing I want you to know is that, yes, cleavage. So from there, we are going to get, let's say, 2 cell stage. Let's say I have this. Now, still, you should be aware that I will still be having my zona pellucida intact. Let me just make this one undifferentiated so that now this time around it has fully divided, so we can see them as 2 cells in here. So for this reason, this zygote over here, so-called zygote over here, this time around I'm going to have what we call 2 cell stage. 2 cell stage. So the nomenclature is 2 cell stage. Yes. Now, one thing I want you to know is that, as I said, as we go through the division, the size of the cells, okay, will be decreasing. But one thing I want to put inside you over here is that the nuclei, the size of the nuclei, okay, will not change, will remain the same. However, the quantity of the cytoplasm will reduce. And that is why we say that the nuclear cytoplasmic ratio of the product of the division. And my friends, one thing I want to put across over here is that the products, these cells we are going to get, will be called blastomeres. Blastomeres. The products, okay, of the division, which we are calling blastomeres, by way of the nuclear cytoplasm ratio, will be increasing. Nuclear size is remaining the same, but the quantity of cytoplasm is reducing. So the nuclear cytoplasm ratio will be increasing. That's what we see. Yes, but still we have our zona pellucida intact. But my friends, the question is, why do we still have the zona pellucida? Yes, the spermatozoa has interacted. Yes, it has helped in that regard. It has also ensured that all those receptors will be removed from it. Okay, ensuring that we prevent something we call polyspermy. I mean, those of you who have not watched the previous video, please kindly get the video and watch it. Now, why is it that it is still in there? It is still intact. The reason is simple. Because we want to prevent something we call ectopic pregnancy. Now, the conceptus over here is not destined to implant in the wall of what we call the uterine tube. It is destined to implant in, of course, the uterus. For this reason, it got to take its time and gradually come to the destination before it can implant. And that is why God ensured that, yes, although this one is helping in fertilization, it is still intact, ensuring that as it rolls on the wall of the uterine tube, it doesn't get the affinity to implant there. So one more function of this zona pellucida is to prevent ectopic pregnancy. Yes. Now, as we move on, what we see is that now, let me just make it simple over here and then put the last one in there. Now, let me make it simple. Now, at what point after fertilization are we going to see this, I mean 2 cell stage coming? So that's the first cleavage division. And studies have shown that roughly 18 to 36 hours post-fertilization, formation of a zygote. That is when the cleavage will begin, so that we get a two-cell stage. Now, normally, then the next one, usually, let's say from, 36, okay, to what we call 48 hours. 36 to 48 hours, these two cells also divide. And so when they divide, we are going to get 4-cell stage. And when we get that, okay, then it means that, that one, yes, as I told you, 36 to 48 hours, then we get a 4-cell stage. Then from 4-cell stage, they also divide, okay, 48 to 60 hours, okay, then we are going to get what we call the, we are going to get 8-cell stage, and then 8-cell stage in that order. Now, one thing I want you to be aware is that the sort of division that we are seeing, okay, it is not symmetrical, or it's not symmetric. In other words, there's not, we don't get equal, daughter cells, equal blastomeres. One usually will be larger than the other. But in most of the cases, usually the first few ones will come, they are relatively almost the same. Why? Because there's going to be something we call, synchronous cleavage. Yes, synchronous cleavage, in the sense that the initial few divisions will ensure that the sides are almost the same, so that they also divide at the same time. So for instance, 2-cell stage will divide at almost the same time, although there will be some slight or noticeable, lag phase, okay, between the two. What is going to happen is that, yes, you are going to get 4-cell stage almost at the same time. Then we get 8-cell stage almost at the same time. But usually one will divide, before the other. And this synchronous, so to speak, cleavage will cease as we increase the number of cell stages. It will cease. Yes. Then it becomes asynchronous. Okay, so that one can divide at this point. Yes, that's what is going to happen. Okay, as the cells, okay, they increase. Yes. Now, this time where there's that kind of fertilization, which we've seen, we can call it, let's say, day 1, or it can even be day 0. Okay, yes, the difference will just be about 24 hours. Okay, but you can nomenclate it, depend on what you want to do. Okay, so that is one thing that we see. Now, at the level, so we get to the 8-cell stage. The 8-cell stage tells us that there has been 3, okay, successive divisions. At that point, the cells, they are not really compact together. They are loosely attached, okay, to one another. Yes. Then beyond that, they begin to compact. So we've seen the cleavage, the divisions are taking place. Then it will be followed, okay, beyond 8-cell stage, there's going to be compaction. These cells are going to be glued together. They have to form some kind of bonds, okay, glue themselves together. So we get, let's say, the 8-cell stage and what have you. Now, the 8-cell stage, now the 8-cell stage, okay, beyond that, yes, the cleavage is still going on. So that we get from 8-cell stage, those cells begin to compact. Okay, so from 8-cell stage to, let's say, 16-cell stage, up to even 32-cell stage. Okay, the cells, because they compact, they form some kind of a ball-like structure. Something we call morula. And my friends, I want to, distinguish this morula from other morula that I'm going to see. I want to call this one early morula. Early morula. Yes. Early morula. So when we ask you how many cell stage is the morula, my friends, your answer is 16. That should be your best answer. But of course, if there's 8 there, 12 there, yes, within this range, okay, will qualify. Okay, so that's the morula. Ball-like, so it means having a large cavity and all those cells are there, okay, compact together. Now, we are gradually entering the uterine cavity. Now, take it out, we are still in the fallopian tube. We are now entering into the uterine cavity. Then the question is, on what day does the conceptus? Okay, now all these stages, they are embryo. You can describe them as being embryo, but precisely we can say pre-embryonic period because you are looking at the week 1 of intrauterine life, first week of intrauterine life. Okay, week 1. They are all stages of the embryo. So for instance, zygote is a form of embryo. Yes, all these cell stages, they are all embryos, which we are calling conceptus, because it's a product, okay, of conception. After conception, that's what we see. Now, so we have, let's say I have about 16 to 32 cells stage, or 8 to a maximum of 32 cell stage. So we are having our zona pellucida. Yes. So at what stage does the conceptus get inside the uterine cavity? I'm not talking about implanting there. And my friends, the answer is advanced morula. Advanced morula. Something we may also call it late morula, because there's early morula. It's still in the uterine tube. Then the advanced morula, which is the late morula, is that stage that this guy will enter the uterine cavity. Okay, so at what stage does it enter the uterine cavity? Then my friends, the answer is late morula. Late morula. So we are looking at the late morula. And my friends, at the level of the late morula, what we see? What we see is that we still have our zona pellucida. And then something interesting happens. These cells in here, they begin to organize themselves into 2 distinct areas. Now, one of them, okay, will be at the periphery. Let me use a different color to demonstrate that. So one of them will be at the periphery. So there will be some cells at the periphery. And then we also have some cells, okay, some population of cells, okay, inside over here like this. My friends, this stage we are talking about late morula. Late morula. So that these cells, which have organized themselves into 2 distinct areas, those ones which are at the periphery, okay, at the periphery over here, we call them outer cell mass. Outer cell mass. And those ones inside over here, they will form something we call inner cell mass. Yes, so we have outer cell mass and then we have inner cell mass. Yes. Now, something that we got to know is that about this is that we will be able to see it pretty soon, no time, is that the inner cell mass, okay, is the one who is going to be the embryo proper, the future embryoblast. Yes, it's going to be the embryoblast. Then the outer cell mass, something will form trophectoderm. Okay, we are going to see that trophectoderm and all that. Now, so then the question is, on what day does this late morula, okay, arrive in the uterine cavity? My friends, your answer is day 4. Day 4. Fourth day is the day that this guy, okay, will arrive over here. Day 4. Day 4 is when this late morula arrives in, of course, the uterine cavity. But my friends, is that the stage for implantation? No, that is not a stage. And so what is going to be the stage, and what are the dynamics which are going to take place? Now, we needed this zona pellucida, okay, the one I've shown in red over here, zona pellucida, to prevent ectopic pregnancy inside the uterine tube. So now, once we are in the uterine tube, where is the destined place for this conceptus to implant, then my friends, then there's no need for it to be present over here again. And that is why there's going to be dissolution of the zona pellucida. Yes, the zona pellucida is going to be dissolved, it's going to dissociate. Yeah, so now let me show you that. So there's going to be that kind of dissociation of the zona pellucida, gradually dissociation of the zona pellucida, okay, so begins to disappear. Yes, begins to dissociate, begins to break down. And my friends, studies have shown that there's an enzyme which is responsible for breaking this down. That enzyme closely resembles trypsin. And you know, these enzymologists didn't want to worry themselves too much because its role, its structure resembles trypsin closely with some slight variation. They decided to nomenclate the enzyme responsible for breaking the zona pellucida trypsin-like protease. Trypsin-like protease. Okay, so that's how they nomenclated this guy. Now, also something happens. This ball of cells having some cells to the periphery, which are calling them outer cell mass, otherwise something we call trophectoderm. Let me just put that one over here, trophectoderm. Then having inner cell mass, which I told you will become the embryo proper, okay, embryoblast. We'll be seeing these things. Actually, I am interested in the cells of the outer cell mass, the trophectoderm. They begin to secrete some product. They will make some secretion, okay, secrete some fluid. And also, while they are in the uterine cavity, some kind of fluid may also invade because there's no zona pellucida impeding it, so may invade together with secretions coming from cells of the outer cell mass. Yes, when this happens, the fluid begins to displace the cells in the center over here, which are calling them the inner cell mass, okay, to one side. And so that's what we are going to get eventually. Now, right now, we don't have a zona pellucida. Let me draw it a bit larger, okay? Now, so what we see is that we still have our cells, which are lying in the periphery, which have made the secretions. And what are these cells? These are the cells of the outer cell mass, which we are calling them trophectoderm. And trophectoderm will differentiate to become trophoblasts. And we will be seeing with the fate of this trophoblasts, okay? Eventually, very important in forming what we call the placenta, okay? So that's what we have. They made the products. And that products, okay, will ensure that that fluid, okay, will displace these cells to one side, okay? They will displace these cells to one side. So assuming the cells have been displaced, to one side like this. When you have such a thing, okay, it tells you that there has been a cavity created over here where it's filled with some fluid, and that fluid provided by trifectoderm, and also invasion from some fluid from the uterus. Okay, so at this point, what you will see is that, yes, let me say I have my trophoblast of the outer cell mass. Then I also have this guy, and this is what we form the inner cell mass, which we are calling it embryoblast. Okay. Yes. And then there's a cavity created over here filled with some fluid, and this cavity, okay, is what we call blastocele. Now, my friends, pay attention over here. We started with the zygote, so-called unicellular, went through some special type of mitotic division, we are calling it cleavage. Okay, beyond the A cell stage, they began to compact, form a ball-like structure, which still underwent some cleavage, okay, which we call it morula. Yes, then what happened that at the early morula, okay, the advanced or the late morula, which now enters the uterine cavity on day 4, okay, which now forms what we call the, I told you that although it's in the uterine cavity, it is not the stage for implantation, but rather will have to be converted to another product, okay, of the conceptus. And this product, what we are saying is that there will be some secretion of fluid by the trifectoderm, the outer cell mass, okay, so that they will push the cells to one side, creating a cavity filled with that fluid. And that fluid-filled cavity is what we call blastocele. Yes. Now, this whole process, okay, forming all these things, the process for us to form a cavity which is filled with fluid, which are called a blastocele, is what we call blastulation. The whole process leading to this formation of this guy, who is bearing this kind of cavity, is called blastulation. The process is called blastulation. Yes. For this reason, the product that we formed over here, where it's having the cavity over here, is known as blastula. Blastula is what we formed over here. Yes. For the process is blastulation. That's what generally we see for animals. But my friends, in the case of human being, case of mammals, we don't want to call it blastula, we want to give it a special name. And so for its reason, this whole conceptus, which we are calling blastula generally, in mammals, and for that matter human beings, we want to call it blastocyst. So my friends, if you hear blastocyst, we are still talking about the blastula in human beings, in mammals. And my friends, the blastula is that form, okay, which will be implanted. So my friends, when we ask you the stage, okay, of the conceptus that will be implanted, it is not a zygote, it's not a 2-cell stage, it's not the morula, it's not the advanced morula. We are talking about the blastocyst. Yes. And why is it possible? The reason is that these cells, okay, still, we are having these cells by way of the trophoblast cells. They will still make some products, make some secretions. And those secretions, they are rich in enzymes, which are going to, break through the wall of what we call the uterine cavity, okay, so that they can implant. We are going to see that. So that's the stage for implantation. Also, sometimes you may need some kind of stem cells for some, therapeutic purposes. Stem cells will be derived from this stage only, blastocyst. Yes. The point is, yes, around day 6, this guy had, sorry, day 4, what we have is that we have the late morula, okay, reaching the uterine cavity. We are saying that it will become, of course, the blastocyst, where is the implantable form. So then the question is, at what stage will the implantation begin? My friends, your answer is day 6. On the 6th day, 2 days approximately after forming the late morula, implantation of the blastocyst, begins. Yes. But my friends, it is not a 1-day process. It will take up to 10 days for full implantation to take place. Yes, for full implantation to take place. Day 10, okay, after, fertilization. Which you can say, yes, it is just like, day 24, okay, after the last menstrual, period of the female. Yes, that's what we see. So on day 10 is the day where there's full implantation. Now, why am I saying this? Implantation, as we talk about, sometimes we treat it superficially. The reason is that in human beings, first, there's going to be something we call apposition. There are 3 main stages of the implantation. The first is apposition. So that this conceptus, by way of the blastocyst, will make contact with the wall of the uterus. That is apposition, physical contact. After that, there is the release of what we call adhesion molecules. Molecules to ensure that after the physical contact, this conceptus will stay there. And so the second process is called adhesion. After apposition, there's going to be adhesion. But my friends, that is not enough. That bond is not enough. It can be dissociated. For this reason, in human beings, God ensured that rather these guys over here, by way of cells of the trophoblast, will make some product, some enzyme-rich product, okay, to break through the wall of the uterus. So it will break through and burrow through the endometrium. That is why there's something we call desensitization. I'll be explaining these things for you. Okay, the glands of what we call the uterus will become very tortuous, will make some secretions, lipids in there, so that it will be ready to receive the concept, to nourish it. Yes. That is what is going to happen. So, unlike in other animals, where implantation may be solution just by contact or just adherence, that's all. In human beings, okay, there's that kind of deep penetration in there, in the wall, so that it will be covered with the endometrium. And at this point, the endometrium will be called decidua. So in human beings, there's complete burrowing of the conceptus, okay, the blastocyst, in the wall of the endometrium of the uterus. And that, okay, that deep burrowing, okay, makes it to be covered with that kind of decidua. Will be covered with something called decidua. Yes, it will be covered with decidua. I want you to put it over here. Decidua. Be covered with that kind of decidua. So I'll explain the desensitization in our videos as we move on subsequently. This type of implantation, I want you to be aware of it. We call it interstitial implantation. Interstitial implantation. So the type of implantation which there's complete burrowing in case of human beings is what we call interstitial implantation. Now then the question is, what point does implantation, take place? What portion of what we call the uterus does implantation take place? I've done all these things for you. Okay, they are there. Where precisely does fertilization take place? Your answer is the ampulla of the uterine tube. Sometimes you may also talk about ampulo-isthmic junction. Ampulo-isthmic junction. Yes, that is also one area that you can give as your answer. Ampulo-isthmic junction or the ampulla of the uterine tube. I told you that the final stage of capacitation will take place at the isthmus, so immediately it's ready. Yeah, so you can fertilize it at the ampulo-isthmic junction, or you can fertilize it in, of course, the ampulla region. There are reasons why these things happen. Okay, I've done all those videos. Can you get them and watch them? Now, where does implantation take place? In the uterus. Now, so your answer is the posterior wall of the fundus of the uterus. Posterior wall of the fundus of the uterus. That's where implantation takes place. Also, something that you may talk about, the junction, posterior wall of the junction between the fundus and the body of the uterus. Now, why are we saying these things? Because when it comes very lower towards the cervix of the uterus, the neck of the uterus, then it may be something from something called placenta previa, a form of intrauterine ectopic pregnancy. We don't expect it ectopic pregnancy because the implantation was wrongly placed. Yes. So my friends, today, okay, we are just talking about the first week of intrauterine life. We began last week talking about some fertilization, okay, and today we looked at, yes, eventually the final stage of fertilization is that once the stimulus has reached of course the secondary oocyte to complete a second meiotic division, which was arrested at metaphase, we form the male and female pronuclei, which will come together and fuse. But as I told you, the fusion is not really complete. So it's like an undifferentiated two-cell stage where it's the zygote, but we are still calling it zygote. And then from there, we go through the cleavage, a special type of mitotic division. And precisely in human beings, we have the holoblastic, form. That's what I want you to know. And then from there, the two-cell stage. Initially, I told you the cleavages will be a bit synchronous, but later it becomes, asynchronous, okay? It's not really happening at the same time. And then from there, eventually we will get what we call, beyond the A-cell stage, okay, compaction begins to happen. Compaction, okay, forming a ball-like structure, which we are having a cavity in there, which we are calling it morula. And morula, eventually, that's the early morula. Then later, it becomes late morula, where, of course, some of the cells will be at the periphery, which are called them outer cell mass, and some cells inside, inner cell mass, okay? Which I told you that the outer cell mass will give rise to what we call trophectoderm, which also differentiate to become what we call trophoblast, capable of making some secretion, some fluid, okay, which will push these cells of the inner cell mass, which will be the embryo proper, okay, to one side, creating a cavity, which I call it, blastocyst. And I'm saying all these processes leading to the formation of this guy, which is the one which will be implanted, okay, the blastocyst which will be implanted, okay, is called blastulation. For this reason, this concept is known as blastula, but in human beings, okay, for mammals, we are calling it blastocysts, okay, so that is what we see. And my friends, the special type of implantation that we see is not just apposition, it's not just adhesion, it is actually burrowing, deep burrowing in there, deep penetration in there, something we call interstitial implantation. But my friends, don't confuse it with interstitial ectopic pregnancy, which will occur in the the uterine tube. You don't have that kind of interstitial part, which may occur in about 5% of ectopic pregnancies. Yes, but we are talking about inside the uterine tube being covered with the decidua. And the decidua are still talking about the endometrium, which has undergone some morphological changes, okay, capable of sustaining pregnancy. So my friends, I'm very grateful for your time this evening. I hope you find this helpful. Yes, go through it. We'll learn about all these important things in our future sessions. I'm very grateful for your time this evening. Have a good night, all of you, and may the good Lord richly bless you. Amen.